Now, how can we assess that our assembly is good or bad? So there are different factors which we can evaluate. We can look into the percentage of the sequences assembled. We can look into the accuracy of contacts and scaffolds. We can look into how good the issue of those repeats have been resolved. And we can further look into or investigate the presence of expected genes if they are there. So sometimes we might have the idea about the genes which are present in different species, the homologous genes. So we can have the estimate of those genes while comparing to some closely related species, and we can test our assemblies. The most common measure of assembly quality is N50. What is N50? N50 is the length of the shortest contact in a set making up 50% of the assembly length. So we take all those contacts that make up to 50%, right? And then amongst them we take the smallest one. So if it is larger, the N50 measure is larger. Obviously larger is the good. Or if we need to add too many contacts into that list, and then in that case we can say the N50 number is low, we might suspect our assembly. So let's try to understand it first. So here in this example we have this N50 measures. So we have the contacts, which is the longest contact is 70 and the smallest one is 5 MB. The total length of this genome is 200 MB. So we are interested into find a list where we can have half of it is covered. So half of it is obviously 100 MB. If we take the first one, that is 70, we add the second one, now it becomes 95, and we add the third one. So now the length goes to 115, the cumulative length. All three, they make up 215. So obviously our N50 is this number, the smallest among us that list. So that is the 20 MB. So this measure is important. It can give you an idea about the size of the larger contacts and the contacts covering up to 50%. So if we are having the larger N50 value, that one is good. Another parameter is we can look into some conserved eukaryotic gene sets. So here in this example, what we see here is we have percentages of CEGs in the y-axis. CEG is obviously conserved eukaryotic gene sets. We take the genes into different groups. So we have a group 1, group 2, group 3 and group 4. So basically it's a discontinuous data. It's not a continuous data. There are actually different 4 groups and we are comparing different organisms. Now for example, we have this C. remini, we have Cavia, we have Loxodonta, Pan, Plasmodium and Toxoplasma. So we see that in group 1, this green one, the Pan, so that has mostly conserved. So this assembly seems like better. The second one is obviously the C. remini, that also doing good. The assemblies like the other ones, they are kind of low in group 1. More conservation in group 2, and it keeps on increasing as far as these two are concerned. They are performing good, and same way the other two assemblies are kind of lower. So number of conserved genes is low as per expected. So these two top assemblies, we can say they may be taken as the good assemblies in terms of this matrix, or by using this parameter. Even the best genomes are not perfect. So C. elegans was published in 1998. In 2004, the last N was removed. Remember guys, when the sequencer cannot do a good base call, it substitutes N. So a presence of N's is not good for a sequence. So large number of N's in an assembly, bad is the assembly. So they keep on removing those N's till 2004. And between 1998 to 2013, there had been identified 558 insertions, 230 deletions, 614 substitutions. I take out this from a book that says like this. So the idea of presenting this thing over here was that even this project, which was actually performed as one of the earliest or one of the important genome assembly projects, still there are some issues. So don't be discouraged. If you are doing an assembly project, you must have huge temperament obviously, and you must have great patience. So it's not an easy task. Let's look into some tips which can be helpful while doing those assembly tasks. So we can try different values of parameters like k-mer size for DBG assemblers, and we'll talk about them in a later section, and evaluate the output Some assemblers can do this automatically. So we can try different parameters, number one, and then we can compare the results. We can try different subsets of data, as we might have our library perhaps might have some issues, so we can try repeat our experiment while using different data sets. We might remove duplicates and homopolymers, and we try different assemblers, because as per a competition organized are named as cage, they say there is no good assembler or there is no best assembler. So try different ones and then in the end compare your results. Don't rely on one of them. Cage is actually Genome Assembly Gold Standard Evaluation. That was the venture of University of Maryland and Johns Hopkins. So what they do is they select data sets associated with known high quality genomes, and then they run a set of open source assemblers. So they take the assemblers, they run on the data sets, they play with the parameters, and then they compare their results and publish in those scholarly articles. So about different genome assemblers, and they are continuously playing with them. They are continuously searching different data and trying those assemblers on them and giving you their reports. So in order to guide you or help you select which assemblers you should use or what tips you should get from them. So it's really good to know about them. So the qualities of the genome assemblies are evaluated by different parameters. We can have good assemblies, and we can have in the end we can try to have or refine our assemblies, but as I mentioned earlier, assemblies are not really easy tasks. They are painstaking and they require a good amount of patience.